Well, let's be gender neutral. This could be like brother chromatids. Okay? Let's just call them chromatids. Chromatids and chromatids. Okay? Yes, 2 chromosomes right there. Okay, stages of the cell cycle. G1, gap1, synthesis, DS phase. G2, cell division, mitotic phase, mitosis and cytokinesis. And then the gap 0, the G0. Somebody asked, I think last time, about like, what about resin cells, right? Well, these are the resin cells, the quiescent state, the quiescent cells, senescence. These cells, they resemble cancer cells in a sense that they refuse to die, okay? But they differ from cancer cells in the sense that these cells, they don't want to divide. They don't need to divide. Cancer cells divide like crazy and refuse to die. Quiescent cells, they refuse to die, but they don't have to divide like crazy. They just are there, resting cells, senescent cells. So that is like the overlap and the difference of senescent and cancer. Senescent cells, they are refusing to die, but they don't have to divide. And cancer cells, they refuse to die, but they divide like crazy. These are the gap 0, the G0, quiescent state. Here you have this, the whole interphase and the mitotic phase. We went over this last time. We mentioned this as well, the loose DNA or the uncondensed DNA and the highly packed condensed DNA. Okay? Interphase, G1, S phase, DNA replication, G2, right here. Then it goes to the mitotic phase, condensed, right? Because it's getting ready to divide. So, really highly packed and organized. This is a chromatids because it's going to give one copy to each of the daughter cells. And that's what you have right here. The G2 interphase, right there. Then you have the nucleolus. Then the prophase, you have the early mitotic spindle. They begin to migrate to the opposite ends. You still here have the nuclear envelope, condensation of DNA. Prometaphase, now you have invasion of the kinetochore macrotubules. These are also known as the K fibers. Kinetochore macrotubules, they are called kinetochore macrotubules because these are the macrotubules that they bind to the kinetochores, right here. These ones, that they do not bind to the kinetochores, but they bind to the macrotubules from the opposite organizing center. Those are known as the polar macrotubules. Okay? And the role for them it is to bind to each other and expand. Once they bind to each other, they begin to expand, and they're enlarging the cells. You also have here these shorter macrotubules right there. Those are known as the aster macrotubules. The role is still undefined or known. Well, some people believe, or scientists believe, that the role is basically to orient the centrosome, the macrotubule center, to be closer to the membrane, right, on opposite ends of the cell. But anyways, that is the role of those macrotubules. And then you have the metaphase where they bind these kinetochores on the centromere of the chromosomes on opposite ends. They are bound to the kinetochores, to the macrotubules. They begin to pull them with equal strength, so they are going to be placed in the middle. Then you have that's the signal, right, to go for the anaphase. Then you have the separase that it will cut the cohesin, that is the cohesin proteins, that binds the sister chromatids. Once the separate cuts them, they begin to separate. Anaphase, that is the checkpoint right there. Okay, make sure that everything is bound. Make sure everything's aligned. Make sure it's middle. Make sure all the chromosomes are bound to the kinetochores and the kinetochores to the macrotubules. Because if they are, then they're ready to separate, so they can separate to the daughter cells. If they are not, then this non-disjunction will take place, where the whole chromosome will migrate to one end, and this cell at the end, it will have one chromosome less. And this one will have 2 chromosomes, or 1 chromosome extra, okay, by carrying this whole chromosome to this side. That's why they have to be that checkpoint, right? These kinetochores are bound to these macrotubules, so they can be displaced to the resulting daughter cells. And that's what they're doing right here, anaphase. And then the telophase, here you have as well the whole condensation of the DNA, and then the nucleus formation, and then you have here the mitotic spindle in this mantle. Okay? And cytokinesis, the division of the cytoplasm. the cleavage furrow. Here you have the microfilaments being playing a role right here. Right here, microtubules, right here, microfilaments. Okay? Right, so here basically the whole mitotic spindle, the one that I mentioned right now. So, keep one. This will be on the exam. Make sure that you are able to draw this or explain this if I show you a picture. Identify the key players on the mitotic spindle. You will say, here you have the microtubule organizing center, that is the centrosome, a pair of centrioles, one centriole, another centriole. That pair of centrioles is a centrosome, and that is known as the microtubule organizing center. MOT, microtubule organizing center, MOC, okay, microtubule organizing center, MOC. And then from here you have these microtubules growing from this microtubule organizing center. These one are the shorter ones. This one are the aster microtubules. They look like a star. That's why they're called aster. And then you have these kinetochores that they will go and bind to the kinetochore. These microtubules that they want to bind to the kinetochores. They are known as the kinetochore microtubules because they bind to the kinetochore. Okay, kinetochore microtubules. They bind to the kinetochore on the centromere of the chromosome. And the role is to bind it, pull, and then put them in the middle, and then move them to the opposite end of the cell. Then you will have these overlapping non-kinetochore microtubules, or known as polar microtubules. They are bound to each other, okay, from opposite microtubule organizing centers. So they're bound to each other, and then they begin to push, they begin to enlarge the cell. Okay? So make sure you know the role of each of those key players on the microtubule organizing center. The mitotic spindle, centrosome, centrioles, aster microtubules, kinetochore microtubules, and polar microtubules, and the role. Okay? Make sure you know that. If I ask you to draw it, draw it. If I show you a picture, if I ask you to define it, what are the key players in the function, then make sure to draw it. Make sure to identify them, which one is which. If I have, like, for example, here, A, B, C, D, then you tell me which one of these. Which one are these labels? Okay. A, let's say the A is here. A is the aster. B is the centrosome, the microtubule organizing center. If I have here C is the polar microtubules, the overlapping non-kinetochore microtubules. If I have here D, this will be the kinetochore microtubules, the microtubules that bind to the kinetochores.
Professor?
Yes.
For the overlapping non-kinetochore microtubules, it sounds like you're saying polar microtubules.
Yes, that is also.
Oh, it is polar.
Yeah, polar
oh, okay.
Yeah. So these one are known as polar microtubules. Polar. These one are known as the K fibers because they microtubules that they bind to the kinetochore. K fibers, polar microtubules. Okay. Yes, polar. Yeah. Polar microtubules, they are going to bind to each other from opposite microtubule organizing centers. Okay?
And then sorry.
Go ahead.
Kinetochore is the K fibers.
K fibers.
Okay.
The polar microtubules, those are the ones that are going to bind, they're going to expand, they're going to bind to each other from opposite microtubule organizing centers, and once they bind, they begin to grow, and as they grow, they are expanding the cell, so then will come the microfilaments to do the cleavage form, and then you will have two salty bladder cells. Mm-hmm. Okay? So make sure you know the role of each one of those. Here's the video. Oh, stop playing. Okay, we're just going to YouTube and see these same videos last time. If we have time at the end of the class, we can watch it. If not, make sure you watch it. Here in this video is really like crude animation about DNA, the nucleopods, and also about the kinetochores. Really important proteins. As somebody mentioned last time about like the mechanical role for work, they require ATP for these proteins that they walk along the microtubules, the dynein and kinesin. They need ATP to do all that, to do all movement, to move the chromosomes. So make sure you watch that. It will not be on the exam, but it is good information. Okay. I will not ask you about the role of those proteins, the kinesin and dynein, all that. That will be for like couple of classes. But I will ask you about microtubules, kinetochores, and microtubule organizing center. This one I showed you last time, the cleavage furrow, that is the contractile ring, made by the microfilaments. Okay? So after the separation of DNA by the microtubules, then comes the microfilaments to make the cleavage furrow, and then at the end you have these other cells. But notice that this cell, the original cell, begins to grow and expand in order to divide at the end. It's not that this cell basically becomes two, but it is going to be enlarged. So at the end you will have two resulting equal to other cells. So that is the role as well of the microtubules, begin to enlarge. Those overlapping non-kinetical microtubules, the polar microtubules, they play a role in that as well. Animal cells. For plant cells, then you have here the role of the Golgi apparatus. They send these vesicles to form the new cell plate. So they send these vesicles from the Golgi apparatus and they're joined together in order to create a new cell wall. So here, microfilaments, here, the Golgi apparatus is what takes a role on the plant cells. Okay. That is in eukaryotic cells, right? Animal cells, plant cells. In unicellular organisms, such as, for example, bacteria, then division of one cell reproduces the whole entire organism. Okay, just for example, bacteria. And bacteria do not use mitosis in order to divide. They use this binary fission. They're basically dividing. That's what they're doing. So it begins here with the origin of replication in their DNA. Their DNA has this section, region on their DNA that is called the origin of replication, and with that it begins to replicate. And because it is circular DNA, then once it replicates, then it begins to grow and expand. And one end, it will go to one end of the cell, and the other end will go to the opposite end of the cell. And then at the end, just it cleaves, it separates, it divides. Okay, so at the end, you will have two daughter cells, and each one of them with one origin of replication. It's really easy, really simple, right? It's not like a linear chromosome like you have in eukaryotic cells, in animal cells, plant cells, but it is a circular DNA. So just begin with that origin replication, it copies itself, and then it just moves, once it's finished, then it moves to the opposite end of the cells, and then the cell just divides. Okay? So that is binary fission. Do not confuse this with mitosis. Okay? All right, so application exercise, we have here an onion root tip, so let's take maybe like 5 minutes to find each of these phases of mitosis, find prophase, so you can find prometaphase, but it's really easy to distinguish between them two from these pictures. We'll see if you can find further condensation. If you find one that is condensed and one that is further condensed, then that's prophase, prometaphase. Find metaphase, all chromosomes in the middle. Find anaphase, chromosomes being separated. And find telophase.
So all four of them?
Yeah.